Okay, and without further ado, I would like to introduce Dr. Sanai.
Thank you, Tracy. Can you hear me okay?
Yes, I can.
Okay. Well, good morning, everyone. So what we're going to try to do is just keep this pretty light and informational and hopefully leave time for questions or discussion. I'll start by just introducing some of the basic challenges within brain tumor therapy and how some clinical trials can help address some of those things. And then I'll leave some time at the end for any questions or discussion about that topic, and then we'll go on. So as most of you know, for most brain tumors, especially if the tumor is malignant or aggressive, there are 4 pillars of therapy, 4 types of treatment that most patients go through initially when we make the diagnosis. It often begins with an operation. As Tracy mentioned, I'm a neurosurgeon. I'm a brain tumor surgeon that specializes in these types of procedures. And so usually the first step is to remove whatever we've now discovered to be causing problems. That procedure is something that many of you have been through, and we can obviously get into more details about that as needed. Following surgery, the next step is oftentimes a combination of a chemotherapy and a medication. A chemotherapy and radiation. Radiation therapy itself is one of the most effective treatments for most brain tumors. And the reason is because oftentimes, as you know, what we see on the scans for a patient with a brain tumor doesn't tell us the whole picture. And there are, in many circumstances, little cells that we can't see on the scan that can still lead to the recurrence of the tumor. So radiation is very effective at not only taking care of the tumor that we can see on the scan after surgery, but even the cells that we can't see. And obviously there are many different types of brain tumors, but the most common type of brain tumor is a glioma. A glioma is a category of tumor that arises from the brain tissue itself. Gliomas are graded 1, 2, 3, and 4, and in adults, the most common grades are 2, 3, and 4. The grade refers to the aggressiveness of the tumor. So the lower the number, the less aggressive it is, and the higher the number, the more aggressive it is. There are other names for these grades. So for example, a grade 4 glioma is called a glioblastoma, which is one of the most common brain tumors in adults. And for tumors like glioblastoma, there is a therapy that has been proven effective to some extent, and that the name of that drug is called temozolomide, or Temodar is another name for it. So Temodar or temozolomide is one of the most common initial treatments that brain tumor patients receive. And some of you may be aware of this electromagnetic cap that some of you or your family or friends may be wearing, or you've seen others wear it. This is called a tumor treating field. It's made by a company called Optune, or rather, I think the company is called Novocure, and the treatment is called Optune. And basically, the way these tumor treating fields work is that you have to shave your head, place these electromagnetic strips that are attached to a battery pack onto your scalp, wear that most hours of the day. And that collective electromagnetic waves actually have an anti-tumor effect. So this is different than radiation in its mechanism, but similarly effective. Now, as most of you know, one of the major challenges for patients in this field is that we do not have a lot of proven drugs that are effective for brain tumor patients. And the drugs we do have their caveats. So some of you may have heard the terms carmustine or lomustine. There are other ways to deliver these drugs through combinations of drugs like CCNU, BCNU. These are all sort of the same category of drug. These are chemotherapies that are developed in the 80s. They're derivatives of mustard gas. So a chemotherapy in a purest sense is really just a toxic drug that is designed to really hurt your tumor or hurt the patient's tumor more than it hurts the patient's body. But obviously, these are difficult drugs to take. They have side effects for sure. They are effective to some extent, but these are older drugs that we don't use so much anymore. Temozolomide we talked about, and another drug that you may have heard of is called Avastin. Another word for Avastin is Bevacizumab. These are the same things. And Avastin is a drug that does not actually have an anti-tumor effect, meaning it's not going to kill tumor cells, but it can have an effect on side effects. It operates almost like a super steroid, where the drug can decrease the swelling of a tumor. It can make the tumor less symptomatic in the patient's brain. So it's a useful drug, but it's not a chemotherapy. It's what's called a targeted inhibitor. A targeted inhibitor, unlike a chemotherapy, is designed to hit just a very specific type of target in the tumor and not just everything around it. But unfortunately, Avastin is not a drug that is going to, for example, prevent the tumor from growing back. Most recently, one type of anti-tumor agent that was approved by the FDA is called Everlimits. Everlimits has another name for it, Afinitor. And unfortunately, this is a drug that only works in a very, very small subset of patients. And those patients have a very specific type of tumor called a subependymal giant cell astrocytoma, which is a long name that we shorten into the acronym SEGA, S-E-G-A. Now, SEGA is really a tumor that occurs in children, and only a small subset of children with a certain type of genetic inherited disease called tuberous sclerosis. So while this drug is very effective for that subtype of tumor, it's really not effective for, or hasn't yet been shown to be effective for patients or adult patients with more conventional tumors. So as you can see, there are not a lot of proven options out there for brain tumor patients. And we can ask ourselves why that is. Well, new drugs today are developed by the pharmaceutical and biotech industries. And these industries really take on incredible financial risk when they develop these drugs, because any drug development can cost $1 billion or more. And the reason that's important is because that means naturally these companies are not going to choose smaller market type conditions to develop their drugs for. In fact, it's very easy to know where new drugs are going to come for, just based on the size of the market for the particular tumor type. So more common cancers like lung cancer or breast cancer have had tremendous progress and new drugs developed for them and those tumors, compared to brain tumors, which is a much smaller market and obviously also a harder target because the tumors are much more biologically complex. Now, this doesn't mean that drugs developed for one indication can't be used for another. And in fact, most of the drugs I mentioned in the previous slide were not developed for brain tumors specifically. Like Avastin, for example, was developed for colon cancer. And then we later determined that it was effective to some extent for brain tumors. Now, the Ivy Center, which is, as Tracy mentioned, a program and center within the Barrow that focuses on clinical trialing and drug development for brain tumor patients, really tries to change this balance of power by making it easier for companies, large and small, that develop drugs to develop their drugs for brain tumor patients and for patients at our center to get access to drugs that are otherwise unavailable to brain tumor patients nationally or worldwide. And the way we do this is pretty simple. We, first of all, try to make it as easy as possible for these companies to participate, which means that we don't ask them for any money. We don't have any financial relationship with them. We just ask them to provide us with their drug, and we pay for everything else through the funding of the Ivy Center, which is primarily provided by the Ben and Catherine Ivy Foundation, which is the largest nonprofit foundation supporting brain cancer research in the world. As I'll describe to you, we develop these clinical trials in a way that really limit the risk to the patient. And there's value to that, of course, because we don't want our patients to have unnecessary side effects. But the other value to that is that companies that develop drugs also want to avoid complications, medical complications in patients on their experimental drugs. Because if a medical complication happens to a patient on an experimental drug, that oftentimes slows down the FDA's approval of that drug. I mentioned that we don't really ask these companies for anything other than the drug itself. And we also don't really keep anything for ourselves when we complete these trials. So intellectual property, which means really the rights to the data and innovations of any scientific project, those things go to the companies because we want them to succeed in developing drugs for the brain tumor patient population, and this helps them do that. Another thing that helps the companies get new drugs out faster is for us to do our clinical trials faster. And so often at many medical centers, a single clinical trial can take 2, 3, 4 years to complete, and sometimes longer. Our trials usually open and close within 6 months or so, which means that we can trial new drugs faster. And of course, we are the largest brain tumor center in the United States. We are the largest drug development program for brain tumors in the world. And as a consequence, we're pretty good at what we do. And that expertise also helps enable these industry partnerships. And these partnerships include companies like the ones on this slide, as well as several others. And these are companies large and small. And the one thing they all have in common is they all have new promising drugs that we think are very innovative, drugs that have never been tried in brain tumor patients before. Sometimes these are drugs that have literally been developed specifically for brain tumor patients, but more often they're drugs that have been newly developed for other types of tumors, but we think they have relevance and value for brain tumor patients. So let's talk a little bit about why it's been so hard historically in the United States and the world to get an effective treatment for brain tumors. The first is that any new drug that's developed first has to get to the tumor, and your brain is naturally designed to keep things out of it as a measure of safety. So most drugs that you and I take, from vitamins to for example, an arthritis medication, these things never make it into the brain. These drugs are incapable of getting into the brain, and most drugs, 90+%, fall into the same category. So the first challenge for any new drug, and the reason that most drugs fail in brain tumor patients, is that the drugs don't get there. No matter what kind of dose you give, your brain keeps the drug out. So developing drugs that can get into the brain is a huge hurdle. The second hurdle is that the biology of these tumors are pretty complicated. And of course, all tumors in the body can be biologically complex. But the fact is that for most other tumors elsewhere in your body, so for example, if someone has breast cancer or someone has colon cancer, these cancers can be modeled, or another word for that is reproduced, within the laboratory. Scientists can develop versions of those kinds of tumors in the laboratory in animals, that look very, very, very similar to what happens in humans. And that's very helpful because when scientists develop new drugs, those drugs can be tested in those animals. And if it works in the animal, then it will likely have the same effect in a patient. But brain tumors don't benefit from that same type of predictivity. And in fact, the animal versions of brain tumors are very, very poor approximations of what happens in patients, which perhaps is not too surprising because the human brain is so much different than an animal brain. But for that reason, it can become very difficult to have breakthroughs in drug development when you can't test it out adequately in an animal and know whether it's going to have the same effect in humans. And a final reason why we find ourselves with few drug options has to do with when and how drugs are taken to clinical trials. And unfortunately, because everyone is so enthusiastic about trying to get new drugs into patients, about trying to discover new mechanisms to treat, the field oftentimes enters clinical trials a little bit early, maybe without all the information at hand. For example, not knowing whether a drug gets into the brain, but still taking that same drug into a later phase clinical trial, hoping you'll see a response, but perhaps never even having a shot at seeing a response because the drug was never going to get there in the first place. So these are the three most common hurdles that face us today. And this is why in my first slide, I only showed you one drug that's typically used in brain tumor patients because we don't have a lot of other options. The conventional way to think of clinical trials is that a clinical trial is when a patient runs out of standard options, when we no longer have anything that's proven to be effective, but still want to do something else. And so a patient enters a clinical trial where they get a new drug, and we give the drug and then see how the patient responds clinically to the drug. And whenever there's a new drug, there are different phases to these clinical trials, depending upon how new the drug is. For the newest drugs, it begins with a phase 1 study. A phase 1 study is a clinical trial just simply designed to determine if the drug is safe. It's not a clinical trial designed to see if there's an effect. It's not a clinical trial designed to determine if one drug is better than the other. All we're trying to find out is it safe to give patients this drug, and at what dose is it safe to give patients this drug? These are trials that can take a year or two. They are very expensive on a per patient level. But if a drug gets through that phase, then it goes on to phase 2. Phase 2 is a smaller scale study, maybe 100 or a couple hundred patients, that is designed to give you some inkling of whether the drug has an effect. It's not definitive. It doesn't prove that a drug is effective. But if you find a drug and you determine that it's safe, then the next step you want to know is, well, is there any chance this could work? And a phase 2 trial is really designed to just tell you if there's a chance. And that can take several years as well, oftentimes longer than a phase 1 study. Finally, if the drug makes it this far, and most drugs don't, after determining that it's safe and determining that it might have a chance to work, then you want to prove that it's actually working. You want to prove that if you give this drug compared to some other drug, that this new drug is going to prolong the survival of the patient, for example, to prevent the tumor from coming back, for example. And so that's done in what's called a phase 3 study. And another word for a phase 3 study is a randomized clinical trial. And a randomized clinical trial is called that because the drug is given randomly to patients, meaning 10 patients might come into this clinical trial, and 5 of them will get the drug, and 5 of them will get a placebo, which is another word for basically a fake pill. And because nobody knows who got what until the very end of the trial, there can't be any bias. and bias is an important thing to prevent in clinical trials. And so it means that at the end of the day, you can see, okay, well, let's unlock the code and see who got which drug, and then let's see who did better. And if the people that got the new drug did better, then that means the clinical trial is positive. And a positive phase 3 clinical trial is how the FDA approves a drug. So for example, with the COVID vaccines, once they did these randomized trials, it was pretty clear that the patients that got the vaccine contracted COVID less often than the patients that didn't. And so therefore that's a positive study. Now, as you can see, all of these phases take years. They're extremely expensive. And it's only at the very end do you really know for a particular patient whether something is working. The other thing that is challenging for the conventional way of doing clinical trials is that we're oftentimes just trying to figure out what is the most of this drug that we can give that's safe and not causing too much toxicity or side effects. So in a phase 1 clinical trial, there's often different dose levels of the new drug. And as you go up in the dose, the side effects go up on the patient. And one of the purposes of a phase 1 trial is to figure out, well, what is the most of this drug that we can safely give that causes side effects, of course, but doesn't cause too many side effects. And so obviously this is a pretty difficult thing to go through if you're a patient on a clinical trial. It's a difficult thing to see if you're a physician leading a clinical trial, but this is the conventional way how drugs are developed for anything in humans. Now, what we do and what I'll describe to you in the Ivy Center and what's called the phase 0 clinical trial approach is we don't worry so much about trying to get so much drug into a patient that it's going to make them sick, because that's not really the best way to get the best effect of a drug. For today's drugs, more drug in your body doesn't mean more effect. There's sort of a sweet spot for the efficacy of a drug in these newer classes of drugs, and it's not necessarily the highest dose. So for example, if you're taking Tylenol and you have a fever and you take one pill, which is 325 milligrams, or 2 pills, which is 650 milligrams, your fever will go down faster with 2 pills than with 1. But for today's newer tumor drugs, that kind of linear relationship doesn't exist. In fact, for some of today's drugs, if you take 1 pill, you're going to have more of an effect than if you take 2 pills. And so this is what's being lost in some of these conventional clinical trialing methods. And this is why we've kind of adjusted the way we do things. So let me walk you through what a phase 0 clinical trial looks like. So phase 0 clinical trials are oftentimes for patients that either have tumors for which there is no cure, or tumors that have recurred after standard therapy, like the things I talked about in the first slide. And it typically begins with us trying to figure out whether there is a new drug combination or cocktail that is applicable to this particular patient's tumor. Are the genetics of this particular patient's tumor a match for the genetics that are targeted by any particular drug? So we go through a screening process. We'll oftentimes get the tissue from a previous operation on that patient's tumor, and we'll test that tissue in our laboratories. And the testing that we're doing here is a deeper level of testing than what some of you may have seen in commercial genetic testing services like Caris, Foundation Medicine, etc. This is a more in-depth view of the tumor and its relationship to potential experimental drugs. And once we find the right cocktail of a drug, that patient will receive that drug cocktail for 1, 2, 3, 4, or even 5 days before surgery. So the reason we're giving them the drugs before surgery is so that when they go to surgery and we remove their tumor, we test their tumor to see whether these drugs are doing something. So the reason we do that is so that we don't have to wait several months to see if a scan is looking better or worse, because we don't want to waste anyone's time taking an ineffective drug. So we can directly measure in a particular patient's tumor, okay, we believe this drug cocktail was going to work. Now let's see whether there's any evidence of that. And if there is evidence of that, then that patient goes on after surgery to stay on that drug cocktail for as long as it works. That might be weeks, it might be months, it might be years. And so this way, only the patients where there is real possibility of the new experimental agent working do they actually stay on the drug long term. And of course, all new drugs have side effects. But that's why we only want to expose patients to those side effects if we know that there is a potential benefit to enduring those side effects. And that's what this phase 0 trial tells us. Now, if the patient goes on the phase 0 trial and the patient gets the drug for, let's say, many months or years, and then at some point that tumor comes back because it has adapted itself to this new drug and can now sidestep its effects, well then oftentimes those patients will go back to the operating room to get the recurrent tumor removed. And that gives us another opportunity to not only put them on another phase 0 trial with a different drug regimen that will challenge their tumor in a different way, but it will let us learn how did that patient's tumor adapt itself to this new drug mechanism, and what can we add to undermine that. So the Ivy Center has these trials running at all times in parallel, and they're constantly opening and closing. So I won't go through specific drugs in the Ivy Center trial portfolio, but I think right now there are 6 or 7 trials that are open for accrual. I will tell you that for some patients, they go through 2, 3 different phase 0 trials as we try to figure out exactly what drug combination is going to keep the tumor at bay permanently. Now, I'll end with just an example of how this works in real life. We talked about gliomas and glioblastomas, which is one of the most common aggressive brain tumors in adults. Another less common but equally aggressive brain tumor is a malignant meningioma. Meningiomas are typically benign tumors that arise from the lining of the brain, not the brain tissue itself. But there's a small fraction of meningiomas, about 5% of them, that are not benign. They are malignant. And they look like this on an MRI. They can be large, they grow quickly, and they're also incurable. And so this young man went through what's considered to be the standard of care for malignant meningioma, which is surgery followed by radiation. And then not too long after, he developed evidence of a recurrence. And you can see that recurrence here, this white spot sort of beneath his forehead. And so at the time of that recurrence, we then put the patient on one of our phase 0 clinical trials with a drug that was a tailored match to the mechanisms driving this tumor. That was over 4 years ago. And to date, that drug is something this patient continues to take, but that drug has been very effective in keeping this tumor from coming back. And because we were able to learn this very quickly in a phase 0 clinical trial, there's now a randomized clinical trial underway to look at this drug in a larger set of patients and get FDA approval for the drug for malignant meningiomas. So I wanted to keep time for questions, and we can do questions now or at the end. I'll leave that up to Tracy. But my name is Dr. Nader Sanai. I thank you for joining us, and please let me know either now or later if there are any questions I can answer for you.
Hi, Dr. Sanai. I do have some questions from our attendees. And my name's Tara. I'm a research nurse with Ivy Brain Tumor Center. So thank you for submitting your questions. So Dr. Sanai, the first question is, how do you accomplish clinical trials so fast? 6 months seems so short.
It's a great question. So the speed of a clinical trial has a lot to do with a lot of mundane details around it. So for example, you can imagine that if there's a new drug that a university or medical center wants to trial, you first have to enter an agreement with the company to get that drug. And any agreement involves lawyers, for example, and contracts. And for some medical centers and neurological institutes, that process itself can take years. But as I kind of described initially, we've really tried to limit any levels of complication in these relationships. So we enter these relationships with the companies very quickly, and we design these clinical trials very efficiently. And then most importantly, because we have so many brain tumor patients coming through our program, we're able to accrue or enter patients very quickly. And research nurses like Tara really help us specialize in this. So it's a combination of efficiency, high patient volume, and of course, willing patients who are interested in trialing new drugs. And then probably most importantly, it's the fact that we're not just hanging our hat on what an MRI 3 months down the line looks like. Because as we all know, MRIs can be deceiving. Sometimes something can look wrong on an MRI, but in fact, it's not an indication of a tumor recurrence. And sometimes a tumor can be recurring and the MRI sort of underplays it. So we depend more importantly on the tissue itself. And by being able to acquire the tissue in the operating room and test the direct effects of a drug, we can get the answers a lot quicker than waiting for imaging. So it's a good question, and the answer is a combination of all of those three things.
Great, thank you. Another question we have here is, my wife has both a grade 4 glioblastoma and stage 2 breast cancer. The breast cancer did not metastasize into the brain. Would she qualify for a clinical trial?
That's a great question. So it's an important question because many clinical trials don't allow you to enter the trial under certain circumstances. So for example, if a patient is too sick to go through medical interventions, then that patient probably won't qualify for a clinical trial. But another important criteria in many clinical trials is whether you have some other cancer somewhere else in your body. And so your wife having stage 2 breast cancer could disqualify her. I can tell you that for most of our clinical trials, that would not be disqualifying. There are exceptions. Some of it depends upon which one we're talking about. But generally speaking, if we can make sure that there's no active issue with the other cancer, meaning she's not going actively through chemotherapy, meaning that the cancer has been relatively stable, then it should not be a problem.
Okay, and our last question, is there treatment for recurrent GBM if operation is not recommended because the tumor is too close to the speech area and the patient is too weak to recover? Right.
So I think this question is getting at the point that for these types of clinical trials, they're only applicable for patients that are going through surgery. And for many patients, they've been told either that their tumor is inoperable, or for some patients, they simply don't want to go through surgery even if it would be helpful. And so the answer is, if surgery is not an option for a patient, there are clinical trials beyond these at our program. And those are the more conventional phase 1, phase 2 type trials where there's no surgery involved and you're basically taking a drug on faith. We're giving you the drug because it's our best estimate that this drug might be helpful, but we don't have any direct evidence of that. And that's the typical way of doing clinical trials. And then of course, there are a handful of other drugs that are FDA approved for other types of tumors that can still be used in recurrent glioblastoma patients. So that's what's called off-label therapy. When an oncologist, and I know Dr. Fonkum will be speaking to you shortly, our chief of neuro-oncology, but when an oncologist prescribes a drug for a patient, even though that drug was not developed for that patient's condition, that's still allowed by the FDA under certain circumstances. So I would say the answer is twofold. Number one, it's always important to get second opinions when you're talking about complex tumors and things in the brain in particular. And so many times when a patient sees a surgeon and that surgeon feels like the tumor is inoperable, they might get a different assessment from a different surgeon. It's important to be kind of dealing with surgeons that operate on these patients routinely. And in tumors in language areas, for example, where language mapping and awake mapping operations are really effective in preventing problems, it's important to consult with surgeons that do language mapping and awake craniotomies. So I would say, first off, make sure that surgery is not an option. And then second, if it is truly not an option, then these other routes will be probably available to her.
Okay, that's all the questions we have. Thank you all for your wonderful questions, and thank you, Dr. Sanai.
Thank you all. I will stick around and be available as this program continues.